Hi, everyone. My name is Alycia Halladay. I'm the co-chair of this session, and I'm also the Chief Science Officer at the Autism Science Foundation. I'm the board chair of the Phelan McDermid Research Foundation. I want to thank my other co-chair, Dr. Shafali Jeste, who's giving the first presentation, as well as our discussant, Dr. Wendy Chung, and the presenters and all the authors on this session named Parent-Researcher Partnerships Across Rare Genetic Forms of Neurodevelopmental Disorders and Autism Spectrum Disorders. And of course, thank you all for joining and having great questions today. This session was actually inspired by collaborative work that is done across different organizations that are taking place already by advocacy groups and researchers in order to better understand diagnosis, make genetic testing more accessible, understand comorbidities, and also leading to the easing of suffering of families affected by rare genetic disorders. Each of the three talks you know about. I'm just going to go over them quickly. The first one is a clinical research partnership with PAGS, and that's going to be given by Dr. Jeste. The second is the role of patient advocacy groups in promoting research and intellectual disabilities, perspectives from patient advocates, and that will be done as a team from Elizabeth Jalazo and Vanessa Vogel-Farley. And the last talk will be partnering with PAGS to advance preclinical models of neurodevelopmental disorders and rare genetic disorders. That will be given by Jill Silverman. And finally, we'll have a great discussion led by Dr. Wendy Chung. Now, I want to talk a little bit about why this session. It's really important that patient advocacy groups, scientific researchers, and that includes patient advocacy groups, as well as other funders, researchers, and institutions, including those that work in industry, to work synergistically and not independently. And why is that important? Well, first, families have the firsthand knowledge of the specific challenges and obstacles that they face when they have a family member with a diagnosis. In some, but not all cases, the researchers themselves, I mean, this is common, but it's not the rule, may or may not even be able to have access or be able to meet a family who's affected by a rare genetic disorder. On one side, the patient advocacy groups don't really know how the sausage is made, essentially. They want to see the discoveries happen, and they don't always understand the logistics and the challenges of getting funding and how hard it is to develop a drug and have it meet a level of evidence-based efficacy. There's also critical infrastructure that needs to be built, and sometimes patient advocacy groups take that on, but the infrastructure itself isn't the most exciting thing in the world. And sometimes patient advocacy groups don't get so excited about this important infrastructure, but they are absolutely key, as you will learn, in better understanding or better building some of this infrastructure. Also of importance are these preclinical models, and you're going to hear about this from Dr. Jill Silverman. People think animals are animals and they're not really relevant to disease, and don't really necessarily kind of even understand the importance of these preclinical models to getting a drug to a clinical trial. So this session is really really intended to promote the crosstalk across patient advocacy groups and researchers across the space, anything from preclinical, basic, all the way to clinical research. And it's a bi-directional relationship in many ways. I mean, patient advocacy groups not only benefit from scientific research, but of course they participate in scientific research. And this research leads not only to treatment, but understanding the disorder, which of course helps families down the road. Now, I can't stop this. I can't end my introduction without discussing Agenda, which is the Alliance of the Genetic Etiologies of Neurodevelopmental Disorders and Autism. This is a group of patient advocacy organizations. There's now about 20 of them. Researchers, we invite industry, family members, and the whole point is so that we are working together and our objectives are aligned. Too often, different patient advocacy groups work with researchers in different silos, and sometimes things are repeated, and as one person said, the wheel gets reinvented over and over again. So the point of Agenda is to not just foster and promote the relationships between patient advocacy groups and scientists, but to do that in a way that moves forward the research to better help families affected with rare genetic disorders. So I'm going to stop my screen share and introduce our first speaker. Our first speaker is Dr. Shafali Jeste. Dr. Jeste is going to discuss her experiences working with the Duke 15Q Alliance to utilize family meetings to better understand the Duke 15Q disorder and to collect data and share data with families. She's a professor in psychiatry, pediatrics, and neurology at the David Geffen School of Medicine at UCLA. And she is also the director of the UCLA Care and Research in Neurogenetics, or Caring Clinic at UCLA. And she's also an important advisor to Agenda. So thank you, Dr. Jeste, and take it away.
Thanks, Alycia, for helping me to organize this. Alycia really gets all the credit for bringing all of us together. And thanks for all the panelists for joining in. I think that, just if you look at the backgrounds and the experiences of the folks on this panel, I think it speaks to the the need and the value in crosstalk and collaboration. We have representatives from basic research, from patient advocacy, from funding organizations, from clinical research to clinical medicine in these rare neurodevelopmental disorders. And it's really critical to have that crosstalk as we make progress in our area of clinical trials and precision health. So what I'm going to do in about 10 minutes, just over the next 10 minutes, is tell you about work that we've been doing in Duke 15Q syndrome and how our partnership with the PAG, the Patient Advocacy Group, has truly accelerated and frankly facilitated our ability to make the strides that we have. And particularly those strides that we've made is in an area that I think of as clinical trial readiness. So to kind of just lay the framework here, we're in this really exciting era in our field of neurodevelopmental disabilities, where as we discover individual genetic etiologies and understand syndromes a bit better, we are developing therapeutics that could be truly transformative from the standpoint of outcomes in these patients. And this is just a slide showing the trials that we just have ongoing at UCLA. We have a couple of phenomenal antisense oligonucleotide trials ongoing in Angelman syndrome, drug trials in CDKL5 and Duke 15Q, as well as a natural history study in Angelman, and the list just keeps growing and growing. So we're in this exciting area where we have the promise of therapeutics in these neurodevelopmental disorders. And it's in that context actually that we've been studying and trying to understand and work with children and adults with Duke 15Q syndrome. So just to give you a quick background on Duke 15Q, so it's defined by maternally derived duplications of chromosome 15q 11.2 to 13.1. That duplicated region is actually one of the most frequently reported chromosomal abnormalities in neurodevelopmental disabilities. There's a gene reviews that was written a few years ago. We just revised it to update it with all our new information. But what's important is that in this 15q region, there are several genes critical for synaptic function and integrity and for really just cortical development. Including the maternally imprinted UB3A, as well as a cluster of GABA receptor genes. And we fundamentally believe that GABA neurotransmission is altered in this condition. There's two types of duplications that I won't get into the details. One is an interstitial, where there's one extra copy within the chromosome, and the other is the isodicentric, where there's two extra copies that are set as a supernumerary chromosome. So it leads to four copies or two extra copies of the chromosome. Phenotypically, these kids have features that are not that dissimilar from other syndromic neurodevelopmental disorders. So these children have hypotonia and motor delays, leading to global developmental delay. As they get older, a large proportion are diagnosed with intellectual disability. Many have social communication deficits as well as autism, and then a large percentage, more than 50%, have epilepsy. Interestingly and importantly, also discovered just in case series of clinical electroencephalograms that were collected in patients is that they have these high frequency EEG oscillations that actually look like a pattern that we see in patients who are taking GABAergic modulators like benzodiazepines. And so this is kind of an interesting EEG finding that I'll talk about in a minute, but it suggests that there may be sort of an EEG biomarker or a pattern in this condition that's worth sort of further exploring. So we've been over the last few years really working towards what I think of as clinical trial readiness in DUP15Q syndrome, meaning we want to better quantify this potential electrophysiological biomarker in a larger cohort of kids. We want to examine clinical endpoints and really determine, what are the measures, the clinical measures that best capture both function and variability in that function in these children. What are the areas that are most impacted in these patients that we might want to try to target with therapies? And by doing so, we also want to really start building a relationship and some trust with the patients and all the families, so that as we develop these trials, we have a clear partnership that's developed from the beginning. And so to really promote this, we've partnered very closely with the DUP15Q Alliance. And so what we did was, as Alycia mentioned in the beginning, is we decided that instead of trying to, force families to come to UCLA, which is hard even if you live in Los Angeles, right? So instead of making families come to UCLA for our studies and endpoints and biomarkers, that we would try to actually go to the conferences. And what we did was we went to 3 different family conferences and collected both behavioral data as well as EEG data. I brought a whole team of folks, and the PAG and all the families were so incredibly important in being able to facilitate that kind of research. But the nice opportunity there is that families are already there, right, to learn from scientists and from clinicians, to meet with each other and to network and to support each other. And so we're really trying to bring the research to them. So some quick snippets of what we did in those meetings is we saw about this is the total sample, 91 participants, which is much more than we probably would have been able to see just at UCLA. As you can see, most were at one time point, some were at multiple time points. And this is across 3 different family meetings. We collected EEG, we did some direct assessments, and then we also actually partnered with the PAG to send, so to do some parent report in person, but also to send electronic questionnaires so we could collect extra data, especially for families who couldn't finish some of the questionnaires in person. So to show you again, just quick data from this, much of this is actually very recently published. So I'm not going to spend the time, since my time is limited, to actually go into the details of the actual data. I wanted to focus more on process. But you'll see here, we really collected a range of assessments at the meetings. We did the Mullen or the DOS, the ADOS, the social responsiveness scale, the Vineland, which is a parent questionnaire, aberrant behavior checklist, and epilepsy questionnaire. So the direct assessments are really on development and cognition, as well as direct testing of autism symptoms. And you can see kind of the sample sizes we were seeing. It really depended on the ability of the child to partake, right? And the developmental level of the child, which assessments we actually did. But we were quite successful in gathering data across a large range of children. To just give you a glimpse of what those data looked like, one of the big themes that came out was that in our standardized direct measures of cognition or development, many of these children do fall close to the floor. And so we actually here calculated ratio IQ scores, so not even the standard scores. And even then we had children sort of in the 0 to 20 range. And I think that was important for us to recognize that these kids have abilities that are real, and we expect to see change in those abilities, but maybe these direct assessments like the Mullen may not be as sensitive to some of those individual abilities that we need to really assess. On the flip side, though, the Vineland Adaptive Behavior Scale, which is a parent questionnaire about daily living skills, behaved much more well, if you will, in this population. We saw a nice range of scores in our kids. And again, the red and the blue is divided by kids who do and do not have seizures. But again, a nice range of scores, and you can see this nice histogram here. So it led us to really support the use of the Vineland in this population. We also know that these children have pretty significant motor impairment. And so with a fantastic junior faculty member named Dr. Rajutha Wilson, that's her here, we actually brought the gait mat to one of the meetings, rolled it out and had kids walk across the mat, and were able to collect some really nice quantitative data on motor skills. And what we found was that, in fact, gait is quantitatively different in children with DUP15Q. They have smaller step lengths as well as lower velocity. And again, all able to be done by actually going to the meeting to collect some of these data. We also collected EEG data at the meeting, and what we were able to do was quantify that EEG pattern that we saw on clinical EEG. And this has been published as well. Recently, we did a second study where we actually tested some of the properties of that biomarker, but quite quickly what we found was that children with DUP15Q have an increased amount of power in the beta range, which is like 15 to 30 hertz. And you can see that on the heat map here, the red being more power, and also on this power spectral density plot. Again, because we were able to test larger samples with the meetings, we were able to then test whether it was stable over time, whether it was scalable, whether it related to phenotype, and all of these studies were really able to be done because of our larger sample. And in the interest of time, I'll skip showing the actual data there, but I will make one comment because this goes back to the partnership with the PAG, is that, as we were studying this biomarker, one of the questions that we had was whether those beta oscillations persisted in sleep. And so as we started looking just at the clinical EEGs we had at UCLA, what we started realizing was that, wow, these kids not only have beta in their sleep, but they have profoundly altered sleep physiology. We're finding reduced spindle density in these kids, as well as reduced slow wave sleep. And so what we did was we partnered again with the PAG to ask, can we collect data from the clinical overnight EEGs that these children are already getting just by virtue of getting their routine epilepsy monitoring? And with that partnership, we were able to gather a much larger sample size of overnight EEGs, which allowed us to quantify the data the way I'm showing you here. And that work is actually ongoing and really requires that partnership with the DUP15Q Alliance. And so I'll just end with like this basic rubric, right? So we think about kind of precision health in DUP15Q in particular, but really in any genetic syndrome, you can think about, if we diagnose a child with, a neurodevelopmental disability and obtain genetic testing that shows that they have DUP15Q and likely altered GABA neurotransmission, this biomarker that we're quantifying, will easily be able to be studied either through the clinical EEG or through a research EEG. As we develop targeted treatments that might, for instance, alter that abnormal GABA neurotransmission, that EEG biomarker could serve as a very rich and important and robust measure of target engagement. And then based on all these studies that we've been able to do, we can then determine what are going to be our most, again, robust outcome measures. And in our case, those would be quantitative motor skills, adaptive skills, maybe sleep physiology, and maybe raw scores for the IQ rather than those standardized scores. So just to finish, take-home points, I'd say that in DUP15Q, a lot of the considerations that we have around clinical endpoints are very similar to other syndromic intellectual and developmental disabilities, such as the floor effects, limits of standardized assessments, motor impairment. But the partnership with PAGS really promotes studies that are large enough and clinically representative enough and diverse enough to be able to answer some of our questions. And I would say that our clinical trials are going to need similar flexibility and creative protocols to ensure successful recruitment and implementation. And so with that, I'll stop and say thanks to all the families. They're incredible. And here's my research team, and I'll stop there.
Great. All right. Hi, guys. I am Liz Jalazo. I am a pediatrician at the University of North Carolina at Chapel Hill and the clinical director of the Angelman Syndrome Foundation.
And I'm Vanessa Vogel-Farley, the executive director of the DUP15Q Alliance and co-founder of the Commission for Neurodevelopmental Copy Number Variants.
Vanessa and I are thrilled to be here today to discuss the role of patient advocacy groups in promoting research in intellectual and developmental disabilities. I want to thank first the chairs of this session, to Alycia and Shafali, for bringing together this fabulous, diverse group of collaborators. I will start us off by going through the specific partnership between the Angelman Syndrome Foundation and the DUP15Q Alliance to develop a clinical research network. And then Vanessa is going to wrap us up by talking about some additional patient advocacy group-led research networks, other examples. All right, so we'll begin with the problem at hand that we set out to solve. And I should say this was clearly not the only problem that we were faced, but a few of the problems that were facing the Angelman and DUP15Q communities. I should also go ahead and briefly explain that Angelman and Dup15q syndromes are two rare genetic intellectual and developmental disabilities, rare neurogenetic disorders that stem from either missing or extra genetic material of the same region, as Shafali pointed out in her initial talk. So this was, there's significant overlap in our basic science and the translational arena. And so this was really a logical partnership for our two communities. One of the most pressing issues in our community was lack of appropriate natural history data. In 2019, the Angelman community was on the cusp of first-in-human clinical trials with potentially transformative therapeutics, but we really needed critical natural history data to inform both clinical trial design and outcome measures. The Angelman Syndrome Foundation had begun funding multidisciplinary clinic sites in 2011. And between a handful of sites that we had active at the time, we knew that there was a mountain of natural history data just inaccessible. One site in particular had been following more than 300 Angelman patients for more than a decade. So there was really, longitudinal data in these academic silos. And more importantly, and perhaps more painfully, as a parent, countless hours of caregiver time spent providing this data and patient time spent traveling to these clinics and participating in clinical visits. So the first issue was really these large siloed data sets within academic institutions that were largely inaccessible. To further complicate this natural history situation, not unlike other rare disease communities, we had multiple natural history studies to contend with. An initial RDCRN funded natural history study, and then a new FDA funded natural history study that was set to start enrolling imminently, as well as a global patient registry that was collecting really critical real world data, but no way to link these rich data sets together and make them accessible for broader use. And then finally, no consensus in standard of care for patients, leading to wildly different management and baseline treatments. So in 2019, the Angelman Syndrome Foundation and the Dup15q Alliance decided to partner in a dramatic expansion of our existing clinical footprints and invest in infrastructure to really harness the power of a clinical research network to streamline data collection, both to inform clinical care, but also to support research. We began by bringing together all the relevant stakeholders, including other patient advocacy organizations, researchers, academics, and pharmaceutical companies with Angelman or Dup15q therapeutics in their R&D pipeline. We wanted to ensure primarily that any infrastructure that we invested in, knowing that this was not going to be a cheap endeavor, would meet the needs of all stakeholders. We hired RTI International in North Carolina to convene this initial stakeholder meeting and help us begin to sort through these data sets that existed and conduct a needs assessment to determine whether what we were collecting currently really met the needs of all parties, including the FDA. And the goal really, as it was conceptualized early on and has continued to evolve, is a centralized data repository that really functions as a linking hub for all of these disparate data sets, with an additional really critical goal of making this data accessible with appropriate oversight that gave patients, parents, and other data owners a seat at the table to decide how their data was being used. So in 2020, the Dup15q Alliance and the ASF launched a combined clinical research network, leveraging the geographical footprint of both smaller existing clinical sites. The result was 20 domestic clinic sites with 4 global partners poised to provide expert clinical care, but also support prospective data collection into LADDER, and then ultimately grapple with this issue of the historical data sets that existed out there. So LADDER, which stands for Linking Angelman and Dup15q Data for Expanded Research, was really developed to maximize the potential of several data collection efforts that were being conducted in both communities. LADDER was designed to promote collaboration with researchers and pharmaceutical companies to advance knowledge of these conditions and promote the development and testing of effective therapeutics. Parents and caregivers first enroll in LADDER to provide consent to link the various data sources on an individual with Angelman or Dup15q. And then we use global unique identifiers or GUIDs along with a matching algorithm to match extant data sets with enrolled participants. Once all this data is linked, all identifiers can be removed from the complete LADDER data set for data curating and sharing. RTI maintains the linkage database on a secure network. And there's multiple ways researchers can be involved with LADDER, both as consumers of the data sets or as contributors to the data in LADDER. And as we've gotten this stood up over the last year, this has been a labor of love for sure. And it's gained more traction. We've started forging really exciting collaborations and really making this data accessible for use in much different ways than it was 2 years ago. We simultaneously began working to bolster the infrastructure of the clinical network to develop a sustainable network to meet the needs of multiple stakeholders. Next slide, please. We implemented monthly learning network style meetings with clinicians to really disseminate the expertise of the FEW to clinicians across the country. We worked together with the clinicians and the patient community to both identify patient and clinician-driven deliverables and a list of shared research priorities. And lastly, recognizing the multiple stakeholders and need for robust data collection through this network, emphasis on support for data collection, knowing this was a big ask of our clinicians.
So our 15Q clinical research network structure was conceptualized based upon a project that came out of the Phelan-McDermid Syndrome Foundation, another patient advocacy group, where they adapted the Project ECHO model to reach the Phelan-McDermid Syndrome global community. The goal of this project was to connect local doctors across the country and the world with a team of specialists in neurology and psychiatry who have expertise in the management of neuropsychiatric problems in Phelan-McDermid syndrome, many of which are common to AS and Angelman syndrome and DO15Q. This project brought forward Phelan-McDermid syndrome pharmacological management recommendations that were developed by the Phelan-McDermid Neuropsychiatric Consultation Group. These logic diagrams describe treatment suggestions for PMS individuals who have PMS and also carry a diagnosis of ADHD, sleep disorder, mood cycling, aggression, and/or catatonia. The successful implementation of ECHO PMS has facilitated improved clinical outcomes for children, adolescents, and adults with Phelan-McDermid syndrome suffering from neuropsychiatric symptoms. While the infrastructure of the 15Q CRN was largely inspired by the ECHO project, it is not currently using the ECHO platform. Our adapted structure provides the means to 1, track patient outcomes, 2, identify future research goals, and 3, further our understanding of the needs of the AS and DO15Q community globally, and bring forward clinically relevant deliverables, as Dr. Jalazo mentioned earlier. Effective patient advocacy in neurological conditions like Angelman syndrome and DO15Q involves a multi-pronged approach. Clinical research is one of the approaches, as we've talked about before, but bench research is another area that patient advocacy communities can play an integral role in, and is necessary for truly translational research towards understanding of the conditions and translating that understanding into treatments. Dr. Silverman is going to talk a lot more about the details about this later, but I'm just going to give the patient advocacy involvement piece. The use of models in genetic conditions has long been a starting point and goal for many syndromic genetic conditions. In vitro model studies permit syndrome and even patient-specific detailed analyses that can be done outside of the whole organism that helps to test and improve safety and efficacy before testing on the whole organism. These models like iPSCs, fibroblasts, etc. are made from human biospecimens. In order to make these models, this is where patient advocacy groups come in. Patient and family samples are needed, and often from more than one patient family. For patient-derived cellular models, the recruitment of scientifically valid sample donors, like the different genetic types we have, can play an integral role in characterization and identification of causal genes and pathways. This is where PEGs can play a really, really critical role in facilitating these model creations. Simple recruitment for biospecimen creations, all the way up to organized biospecimen collections at our family conferences or advocacy events, as both Angelman Syndrome Foundation and DO15Q Alliance have done. So let's switch over to the in vivo experimentation that's done in a whole organism rather than in live isolated cells. Common in vivo models, as Dr. Silverman will talk about later, each has their own role and utility in genetic conditions. Supporting the development of models can be accomplished in several ways. For example, the creation of foundation grants programs to fund researchers to enable dedicated research into the genetic condition, and letters of support for researchers who are going in for funding other funding sources in your disease area. But as with every model for any genetic condition, the purpose and utility of the needs need to be anchored in human expression, sort of like through the clinical data that we're collecting through the clinical research network. All models, in order to be useful, need base outcomes and outcome evaluation in the human translation and translation of the human condition. Symptomatology, behaviors, medication usage is where the collection of data facilitated by PEG can really come into play. As Dr. Norbert Weiner said, the best model of a cat is another cat, or preferably the same cat. Since we can't do that in humans, there's no perfect model, but linking the model data back to what we learned from patient data, like the data collected in our 15Q ladder or in any PEG database, is key to unlocking the promise of in vivo and vitro models. So really what patient advocacy groups can do is bring it all together. Patient advocacy groups are very, very valuable providers of insight and input and access to the patient community and data. Patient advocacy groups, above all, are stakeholder conveners, bringing together their patients, communities, clinicians, researchers, and disease models with the goal of ultimately being able to include industry and other therapeutic creators amongst these stakeholders. Thanks.
Okay, great. I will just, let me get my screen shared. And can you guys see the slides okay?
Yes,
Yes.
looks
Okay.
great.
Okay, great. Hi, I am Dr. Jill Silverman. I am an associate professor at the Mind Institute in the Department of Psychiatry. I've been studying preclinical models and neurodevelopmental disease my entire career, so I'm extremely familiar with this area. And I've also been working with parent advocacy groups for quite a while. Initially starting from my research in autism spectrum disorders, but as genomic testing increases, and we've found many syndromic genetic causes of various neurodevelopmental disorders, I've shifted my focus to that area. And so today I will talk to you about what my lab does, which is preclinical models of NDDs with rare genetic or with a rare genetic cause, and how I've partnered with many parent advocacy groups to move our research forward. And so I'd like to start just on a really positive note, which is, because that's not so much how scientists think nowadays, we're very much on the not positive notes, but a really positive note is that in my lifetime, gene therapy is now a thing, something that I thought of as, or that we might have thought of as science fiction is no longer science fiction. And particularly in recent years with the approval of a gene therapy for spinal muscular atrophy and giant axonal neuropathy, we believe that gene therapy could be or will be realistic for many of our single gene disorders. And so it's not all woe is and no cure. And I think we're allowed to use the term cure. I use the term curative to be safe. And why do we focus on these rare genetics when, why rare when not large? And the truth is that it's all going to inform the greater good, right? And so if we understand problems or the downstream mechanism, or can treat what goes wrong when one gene that goes wrong in Angelman syndrome and DUP15, as you just heard from Vanessa, it's the same gene UB3A, or if we understand like how we can fix X-linked disorders such as Rett syndrome and CDKL5, if we are able to treat or to reverse the mechanism of these rare disorders, it's going to help us re-engineer our technology so that we can alleviate or treat broad disorders that have similar mechanistic etiology. And then that goes as big as autism, which is 1 in 59, and numerous intellectual disabilities, 1 in 6. And many, almost exclusively all of these disorders are comorbid with autism and epilepsy, which is our area of interest. And I think the scientific community has kind of come to a consensus around this too. The data from these rare disorders is going to help us build this foundation of understanding, especially as genetic testing becomes more affordable and insurance covers it. I've met many families from the Mind Institute or other places that years ago considered themselves having an idiopathic autistic child, and now they are aligned with one of these or another specific group. And where I come in, like what our role is like I said, I've been studying preclinical or rodent models, if you want to call them that, or cell models my entire career. And that's because of like what they call this, the failure to translate, or like lost in the valley of translation for drug development. It's because there's so many compounds in research and development at drug companies or, even gene therapies at academic and biotech and pharma, but we have to test them before you can put them into a patient. And that is where I come in, the preclinical research, so that we can get to clinical trials. So as Shafali talked about clinical trial readiness, our lab is sort of like the go, no go of testing compounds on a cellular basis and in a living system basis. And so a few years ago, I decided, even though I was trained as a behavioral neuroscientist, that I was going to go down the cellular path. And I teamed up with my close colleague, Kyle Fink, and he started to teach me about iPSC cells, which, as Vanessa described, is a great role for patient advocacy groups, because they can, at meetings, we can collect fibroblast tissue right from the patient. And then that way the disease that we grow in the dish is actually the same genes of the patient, which may have the exact cause. Shafali talked about many of these conditions are lumped under a single syndrome, but they have a few different types of causes. But there is a con into these iPSCs, in that they don't always behave and they are hard to grow, and you do have to sort of get them from a live patient. And that some of the iPSC models that are going around the world for some of these rare disorders have been created from just 1 or 2 patients, which is a definite negative, with not allowing for heterogeneity. This other way that we have been doing it is like we also like test therapies this way, because we grow the iPSCs and then we create a CRISPR isogenic control for that model, and we test a bunch of in vitro screening. And that has been really cool and really productive. And so our groups try to do this in parallel with animal modeling. However, it was really striking for me to find out that, as a person that works with live animals, that getting these cells to differentiate and to grow into neurons, it actually takes about the same amount of time that it's going to take me to breed 2 animals together and come up with a whole animal colony with the same gene mutation. And so Dr. Kyle Fink and I have spent a lot of time weighing the pros and cons about whether we are going to try a therapy that he's designed in the lab and bulked up at GMP grade, if it's worthwhile to even put into the cells when it takes about the same amount of time to make the animals be ready for testing. But what has been a hurdle for animal models for a long time, and I've had the great fortune to see this unfold as I've been in the field for a really long time, from when we had no construct valid animal models of autism to a glorious reduced amount of riches, where we have all of the genes from all of the genetic studies to come out one by one, those animal models. So there's too many animal models. At the same time that clinical trials, and the sad part is that actually the same 2 therapies are the only 2 therapies that have been approved in that amount of time. So that's over 20 years. And what's come out of that is that there's been a real need for improved clinical outcome measures. But what myself on the neuroscience side have also noticed is that there's also a need to improve how we preclinically assess the behavioral models. Because if we're not engaging the similar circuitry or analogous meeting, like what does it mean when animals bury marbles, or what does it mean when they're running on the rotarod, we might not actually know what we're testing, and we may be pushing something forward that's going to fail anyway in the end, or vice versa, not pushing the right things forward. So I'd just like to highlight some examples of how our laboratory has gone through preclinical testing and models. And again, we do this in parallel with the iPSC testing. So you heard Shafali talk a lot about gait already in DUP15 and Angelman. And so since we also study DUP15 and Angelman, I'm going to talk about this as well. We've really been focused on motor as a translational outcome. The circuitry is the same in the brain. It's a really robust, rigorous phenotype. It's quantitative, it's reliable. And there's lots of different ways to get at it. So what Shafali showed you was some data on a gait map, which is called a Xeno Walkway. These slides are all from Jessica Duas, who's definitely a part of this group and has been my great collaborator. I've like really enjoyed working with her. We got a grant together this year to study this. Using integrative gait analysis in Angelman patients. She also does some EMG, and we use this device called an Actomyo, which is like a little Fitbit for the Angelman kiddos' wrists or ankles. And then what an integrative gait analysis would look in a mouse model would be a little treadmill scan with this video camera underneath it that takes the contrast and the pause and gathers about 216 metrics. And this is what an AS mouse looks like on that same treadmill at the same speed with all of the same metrics. And going through Jessica's findings in the clinic and then going through our findings, we were able to find spatial metrics such as the stride length and the stride width and the stance width. And I mean, we have a whole paper on this submitted, but there's about 20 different indices that they just differ on. And then also actually the temporal dynamics. So the way the animals move, the way their propulsion, the way they brake, the way they actually stop to balance themselves, it's different in Angelman kiddos, it's different across development, and it's also different in the animal models. Another thing that we do that's really analogous is use iPad tasks. These have been around for a really long time. They were modified from the CAN tab. This is a study from my friend Jeste, who had done this work in Australia with schizophrenic patients and working memory tasks. And she had this working memory task, and she also got the Road-induced working memory task. It's really amazing. I've had the pleasure and honor to work with colleagues from the Wine Institute like David Hessel, and also work with Audrey Thurm on designing various tasks that could be done by both a mouse and human on the iPad. We've done this in Angelman syndrome. This is rat model data to show that they have clear cognitive deficits. And then this is the CDKL5 model to show that they also had deficits. I just want to highlight that in the Angelman syndrome rat, the reason we had to use the rat was because their motor is so impaired, as you just saw on the gait, that they actually have trouble in the touchscreen task. And Audrey and I have had substantial conversations about the fact that this is common in clinic as well. Okay, one more slide, Alycia. But it's not letting me move. Okay, and so the last thing I want to talk about is EEG, and Shafali talked about this a little bit too. The way we collect EEG, and this is very translational, is we do it wireless, and we use this little telemeter on the side, and there's a screw lens on the cell, and they live in their home cage. It's not a field recording, or they're not tethered at all. We are able to collect so many metrics, such as just spike density and spiking and firing. And this is like what our Dupe 15 looks like, but we're also able to collect power spectral density that Shafali talked about a little bit, has been shown a lot for Angelman syndrome with this like elevated delta power, and Shafali and her team have shown a lot in elevated beta power in Dupe, and we see that the models totally recapitulate or have analogous findings for that. And then sleep, as Shafali mentioned earlier, we're also, because we record for a long time, able to measure this time in all of these stages of sleep, whether they be active. This is active, wake, and moving. This is just wake, slow wave, etc. And just this year, we were able to finally write a Python algorithm that automatically detects spindles. And so we just published this and did it for our Angelman study, where we found less spindles. And so those are just a few examples of the power in preclinical models. And with that, I will wrap it up. This is my team. Nicole does all the EEG, Stella does the gait, Liz does the rats, and Anna's been doing 3 different genetic studies. And definitely want to thank all my clinical collaborators, which is Audrey Thurm, Jessica Duas, Shafali, Jeste, and Ann Anderson. And of course, all the patient advocacy groups, Dupe 15, FAST, CDKL5, Bailey-McDermott syndrome. I wouldn't have been able to come up with those outcomes if I hadn't met so many of the children. So thanks.
Thank you. Thank you all for great presentations. We're now going to switch to the discussion. And I'm going to share my screen, but first I want to introduce our discussant. Her name is Dr. Wendy Chung. She is one of the busiest people in the world, and we're lucky she's doing this discussion for us. She's the professor of pediatrics and medicine at Columbia University. She's a board certified clinical and molecular geneticist and runs a clinical genetics program at New York Presbyterian Hospital and Columbia University. And if that wasn't enough, she is the director of clinical research at the Simons Foundation Autism Research Institute. So thank you, Dr. Chung, for providing your insight for the discussion. We did generate a few discussion points, which I will share with you right now.
So let me just start by saying, wow, what a tour de force. You guys are amazing, each individually and of course collectively as you've worked together. And as I was listening to you say how you've done this with your individual conditions and with your family groups, I was thinking to myself, so how do we do this times 1,000, right? So in terms of thinking about this, how do you scale this? And for many of the things you're doing, they seem to me quite generalizable, not necessarily condition specific. So do you guys have any ideas? How do you do, maybe not times 1,000, let's start out with times 100. How do we do that? Who funds it? What infrastructure do we need, do we use? But how do we take what you've done and clone it for all those other groups out there?
Well, I can jump in here a little bit. I'm the co-founder, as I said before, of the Commission on Complex Copy Number Variants. It's a much longer name. We call ourselves the Commission, essentially. And Wendy's actually one of our advisors. And it was through the CZI Rares 1 grant where we decided to say, how can we do this across disorders? So we partnered together with 3 other complex chromosomal abnormalities, meaning that it's not the smaller, like we don't say that the other ones are less complex because they're all complex, but it's more these other chromosomal abnormalities that have structural differences, and like kind of looking at them across the phenotypes that we're seeing versus the genotypes, and how can we look at that both clinically and then also genetically. And so we've been meeting with different working groups around how do we look at them in models, and how do we look at them in humans, and how do we collect better clinical data, and just kind of coming up with a roadmap and that we can give to other patient advocacy groups to say, how do you do this well? How do you get it funded when you're doing your, whether it be the grassroots fundraising or the bake sales or even the NIH grants that some of these groups are coming into. So it's one of those things. How do you, making it generalizable is also just making the information accessible to patient advocacy groups as they're up and coming is one of the big things that we're finding in that space.
Okay, that sounds great. So I heard there are some very forward-thinking philanthropic groups to fund, NIH to fund. That's useful infrastructure. Any other ideas?
So yeah, I was going to say that FAST is also doing something similar to that Vanessa described. They just formed a larger consortium called NSYNC, which is going to teach, the theory behind it is to teach how they roadmapped, because we went from 0 clinical trials in Angelman syndrome, despite having an animal model for 20 years, to now 3 this year. Of different antisense, and then we're about to, we almost have our IND approved for another type of viral therapy. So they are forming a larger group called NSYNC, which is supposed to disseminate the knowledge into other rare disorders that are sort of just forming and are just getting their feet wet and learning all about the challenges of working with academia and drug companies and partnerships. And then personally, what my lab is doing as far as the EEG and sleep coding is definitely making everything open access. And so all the codes go onto websites. And for a lot of labs, like, you know, we only have so much capacity, so we can't take on everybody's model and everybody's everything. And so it's definitely like, if you have this equipment here, we'll share everything type model.
Let me ask a related question. So as we're thinking about clinical trial readiness, how do we actually do that in terms of hearing what the priorities are from families and thinking about some of those measures we might need to see if our trials or our treatments are actually effective?
Yeah, I think it's a great question. And I was actually, sorry, I was like trying to unmute myself. I couldn't find the button for a second, but you'd think after a year of Zoom, I'd be great at this somehow. But no, I mean, Wendy, I think you're right. And I think, you know, I showed the example of Do15Q, but I mean, not just the clinical aspects of these conditions, but even the processes are so relevant across these various, you know, syndromic neurodevelopmental disorders, as you're saying. And I think that's true for this issue of clinical trial readiness overall as well, is that, you know, what we need is we need large enough samples where we are testing clinically representative, you know, in a clinically representative way, this group of kids and adults. And we need to test, you know, the domains that we know are most likely to be affected in a way that is, you know, clinically relevant and scalable, ideally. Because I think even if we have this dream of, like, you know, hitting the pavement and going to all the family meetings and doing kind of what we did with Do15Q, you know, that's probably not totally feasible. I mean, it would be great to sort of get some gold standard data that way, but I think what we really need is collaborations around these, you know, remote assessments and parent questionnaires and wearables, and leveraging the clinical data we already have, like the clinical overnight EEGs, which you and I have talked about, you know, I think several times. You know, most of these syndromes, most of these kids with genetic syndromes are at high enough risk for epilepsy that they have routine EEG surveillance at some point. So how do we, you know, bank those data and then, you know, pull them to be able to ask the questions that we want to ask around biomarkers or sleep or whatever it might be. And so I think, you know, those sorts of collaborations, I think, are very feasible. And I actually think COVID, in some ways, one of the few perks of COVID was that it forced us to have to test some of these assessments and measures remotely, right? But I think, you know, we just need, you know, we're not going to ever get away again with having, you know, data sets of 10 or 15 kids and trying to make generalizations about clinical endpoints based on that. So I'd love to see more cross-disorder collaborations, just like what 15Q has done. I mean, I think it's really incredible. And I've enjoyed being a part of that process as that collaboration has grown.
Just as another thing that we've kind of partnered really well with Dr. Jeste in that aspect. I think there's a little bit of there's a kind of a call for transparency when it comes to some clinical trials too. Like, we just embarked on our first one in the last 18 months. And there was a lot of questions around there that we had thought we did a good job on clinical trial readiness in terms of educating our community, but there were still some things that we had stumbled over. So we taking that and back to the groups that we are working with in terms of pharma, it's that transparency, that's that openness and that flexibility in that communication that's really making the difference in these places where it doesn't seem like they're doing the status quo. They're not doing the same old. There's some definite integration in with the protocol, like the protocol creation, where they're asking for patient input that it's not the leaders of the organization, it's regular patients who, you know, might come to a conference once in a while, but other than that, they just are part of the alliance because they're connected with other families. So that's been really helpful in our case for clinical trial readiness.
And I'll just jump in and add, I think that that's a critical role that patient advocacy groups have to play. I think they have to serve as that liaison between industry and the patient communities. I think, you know, I've seen countless times over the last, you know, 3 or 4 years that I've sat in this role as the clinical director, you know, how informed an industry sponsor was by those interactions with the families and with, you know, focus groups convened by the patient advocacy groups. So I think that that's a critical role that the patient advocacy groups will always need to serve.
That's great.
And one quick, very concrete example of that is, you know, the clinical global impression, or the CGI, which is, you know, one of our most you know, tried and true clinical endpoints and trials, which is a Likert scale of improvement, you know, with either with time or with a therapeutic. And the Angelman syndrome group, you know, recently, you know, through partnerships with not just clinicians, but with parents and caregivers, developed their own Angelman specific CGI with the domains that were Angelman relevant, with anchors that were incredibly specific to the needs and the challenges that kids with Angelman have. And that CGI is now, you know, hopefully and will be used for trials. And what's interesting is that, you know, as we've been working, and I didn't talk about it too much today, but, you know, we've been working on a clinical trial for Do15Q, which, you know, we hope will be, you know, will be live by the end of 2021. And we talked about, you know, sort of going through that same process with the CGI for Do15Q, which we are going to at some level do. But when we actually went and looked at the CGI that was created for Angelman, we realized there's so many similarities actually, and we could really just modify that and use it for the Duke 15Q group as a start. And we realized that it was blood, sweat, and tears with the Angelman group for years, right, to create that CGI with all the focus groups and everything. But that CGI is going to provide so much benefit for other patient advocacy groups and for other patient communities, who are developing trials and don't have time to wait 2 years to create their own CGI. I think that was just an example.
Yeah, I just want to add in on that, like that fast, like Alison Berent would say, like not only did they make a CGI specific for AS, but that's probably the third. So like they have a Bailey that's specific for AS, and they've modified a lot of scales over the years through their biological outcome measures subgroup, and like Alison Berent would definitely call this clinical trial as flying the plane and building it at the same time so that other disorders don't have to.
Very good. Why don't we pause there and take some questions?
So thank you. I think we're going to turn this over to the general question live discussion. And so we hope you come up with your own questions, and they can be about anything that we've discussed here or some other question that you want to talk about.